I'm now going to introduce our second speaker. So our second speaker is Raji Midori. She got her PhD in biochemistry at the University of Würzburg in Germany, and she's currently a postdoc in the biochemistry and biophysics department at the University of Rochester Medical Center. And today she will be talking about ribosome profiling.
Hi, thank you, Morgan, for introducing. And once again, I'm Raji, and I'm pretty new to United States. I just came a couple of months ago, and I started as a postdoc in Dr. Shinley's lab. And his lab works majorly with this technique, ribosome profiling, and they work on male reproductive system. They work on the model organism they use is mouse. And they do this technique tremendously in this mouse testis to study various pathways, or particularly for understanding protein synthesis that helps for the spermatogenesis, that is the production of sperm. So I'm pretty new to this technique, as you all are. I will try to explain and as much as, in a simpler terms, so you get the concept, what it is, and how good it is as a tool to study different questions that exist, open questions that exist in this field. So here itself, the title, it says it's a monitor for protein synthesis at the genome-wide scale. So you can study within the cell or in any tissue what is happening and what is happening to the protein, how they are synthesized, the rate of their synthesis, and what kind of proteins are expressed. Now, I just want you guys to imagine, this is something really a bigger perspective, right from the, when the life on Earth has started, that is 3.5 billion years ago. It all started with a single cellular organisms. And now you see today where we are, multicellular organisms with diverse functions, and look at the species diversity. Imagine how many species we have, how much diversity there is. So all this contributes to the uniqueness of the genes, of course, they have, but also, if you look within the species, if you look, all of us, we all started from a single fertilized egg, all of us. Now look, we all are different with different, everyone is unique. So even within the species, to come from one single cell to become a fully functional adult with different tissues having different protein expression, and their functions are different. So what is that? That contributes to this uniqueness and also the diversity. So for me, if you ask me what is it, that I would say that's the regulation of gene expression. Of course, the DNA, that is the genes, those are unique for every species, but also to have diversity within species, we have the major thing that contributes is the regulation of gene expression. Even though you have genes, it doesn't mean you have the same protein expressed at the end of the day. So it's like what is getting expressed and at what time and how it is getting expressed. All these matter to the diversity and the function of different organs within our body. So I'm sure you all are aware of central dogma of biology, where from DNA, by a process of transcription, RNA is made, and from RNA, the process called as through a process of translation, proteins are made, and all this process, they regulate, can be regulated at different levels, leading to differential gene expression, and that ultimately results in traits and different cellular functions. Now, at every level, even at the DNA level, different modifications exist on DNA that regulates to the expression of genes. They could be epigenetic regulation. And then even at this level, the transcription level, you have various proteins and RNAs playing to regulate what kind of genes should be expressed depending on the environmental stimulus, or also depending on the age, the growth, the stage of the development. And RNAs apart from the coding RNAs, those are that synthesized proteins, there are also a lot of non-coding RNAs. These non-coding RNAs, they don't really produce proteins, but they really help in regulating this whole process and leading to the protein expression. So then the next is the translation, is the process where the messenger RNA is translated into proteins. And that's, even at this level, there is a translational regulation happening within the cells. And finally, once the proteins are synthesized, that doesn't mean that the protein is going to be active and it is going to give a function. You still have environmental stimulus to keep some proteins inactive, some needs to be modified, or they could be degraded if they are not required. So at every level, we have such a what you call the background safeguard system regulating. It's constantly being in process to, at the end result, whether the protein is really required or not. All these steps are playing a role to lead to a protein synthesis. So it's a very complicated, intricate, safeguarding process to get a good protein expression at the end that is required in correct time. That's important. So now the ribosome profiling, it's a technique that helps us, is a tool to understand the regulation of the translation. So when how the proteins are synthesized from the RNA and the rate at which they are synthesized, and also to have at any given point, what are the proteins those are synthesized in that particular cell. So have you guys ever heard of a ribosome? How many of you are familiar with this term? Okay, good. So just to give a brief introduction, what are ribosomes? These are macromolecular machines. Within a cell, there are a lot of macromolecular machines, not just individual proteins, individual RNAs. They usually don't act alone. They're always, in complexes, different proteins, different RNAs, they form different complexes to achieve a function. So ribosomes are also such macromolecular machines. They're also ribonucleoproteins, which basically means they have RNA and proteins in them. And almost the constituents of this ribosome, 2/3 of the ribosomes are made up of ribosomal RNAs. These RNAs are non-coding RNAs. They don't code for proteins. The ribosomal RNAs, these are the building blocks for the ribosomes. And 1/3 of it are made up of various proteins. And ribosomes are ubiquitously expressed. In every single cell, you have ribosomes. And the simplest way to describe, they have a large subunit and small subunit. If you look at the prokaryotic ribosomes and eukaryotic ribosomes, they are different because they have different RNAs and also the protein components. So if the prokaryotic ribosome, which has 50S large subunit and the 30S, this comes basically from how they sediment in the centrifugation, the sedimentation coefficient, that is. So the prokaryotic ribosomes, they have 23S, 5S RNAs along with 34 other proteins. So it's not just single protein RNA complex, it's a multi-protein RNA complex. And even eukaryotic is much more complex than the prokaryotic one and also achieves much complex functions than the prokaryotic one. So we have 60S and 40S with different RNAs and proteins which make up the eukaryotic ribosome. Now where they are present, if you look at a typical animal cell, ribosomes, if you look in the cell, they are basically present on the rough endoplasmic reticulum, that's the nucleus. This is the cytoplasm. So the nuclear membrane extends into endoplasmic reticulum, and ribosomes are present on this rough endoplasmic reticulum. It's called rough endoplasmic reticulum because ribosomes are bound onto the endoplasmic reticular membrane. You also find free pool of ribosomes. So basically, a lot of translation is happening at the endoplasmic reticulum and also in the free cytoplasmic pool. So now what is the function? So you all know the ribosomes, these are the ones that translate the genetic information that is stored in the messenger RNA into proteins. Now, I just want to briefly walk you through the process of translation, how it happens. So here is the template. The template for the protein synthesis is the messenger RNA, that is the coding RNA. If you look at the template, that is this one here, it has a 5' and 3' end. Here, basically this picture is only depicting the coding sequence. But otherwise, if you look at it, also has a leader sequence at the 5' end, which helps the efficiency of binding the ribosomes onto the template. And also you have at the 3' end untranslated regions where basically ribosomes are not present in there. So ribosomes start at the start codon of this messenger RNA. They bind, they recognize the start codon, and the ribosome assembles on the coding RNA. Now, once the ribosome is assembled at the start codon, then it gets translocated. So you have the codon periodicity, 3 nucleotides. So it goes on adding the amino acids which are required to make a polypeptide chain. That is the backbone of any protein. So you have, it's a multimer of amino acids. That's the protein. So here you see the polypeptide chain growing. As the ribosome is translocated, you see the peptide is getting synthesized. So once this process, it's not like one ribosome binds and it translates the whole region and then the second ribosome binds. It's like happening sequentially one after the other. So you see here the monosome, and once it translocates, the next one binds. This translocates, and then, so there is continuous production from one transcript, one messenger RNA. You have multiple proteins being synthesized. It's being translated multiple times. So once the ribosome reaches the stop codon, which is almost close to the 3' end of the messenger RNA, the ribosome dissociates, leading to the, even the protein that is synthesized fully, it gets released into the cytoplasm. Now, so I just want you to be familiar with these two terms. So when the transcript is there with one ribosome attached, it's called monosome. And when you have multiple ribosomes on this transcript, you call them polysomes. Okay. Now so it's again, the technique, I'll just give you the brief overview of it before we go into the technical details. Ribosome profiling reveals what, when, where, and how of protein synthesis. And it also involves deep sequencing of ribosome-protected mRNA fragments. I'm sure you guys had, if you have attended the RNA sequencing talk, which was there earlier, previously, you must be familiar with the sequencing technologies. Those are involved in sequencing the RNAs and DNA. And it's currently one of the very unique and powerful or single tool that exists to understand what is happening globally for the translation inside the cell. It's a single tool that really, I think a lot of people are using this tool to understand the protein synthesis. So this technique actually facilitated discovery of regulation of gene expression underlying diverse and complex biological process, and also it has led to important aspects of mechanism of protein synthesis, a deeper understanding of how proteins are synthesized inside the cell, and also a systemic approach for experimental annotation of new coding regions. There are a lot of unannotated genes in the genome. So with this technique, you know whether these genes are really getting translated, or you can also annotate this is a new coding region. Now, going into the technical aspects, so if you want to do ribosome profiling, what you need the kind of cell you want to work with. You take those cells and you lyse them first. So importantly, here, you need to capture this state within the cell. What happens, this is constantly happening, so the polysomes are getting released. If you don't stop them here, probably you won't capture this because they are, by the time you lyse, they fall off. So you use an inhibitor to capture stop this elongation process. So you have a stage that is halted with a lot of polysomes on it. So you lyse under those conditions using an inhibitor, and so you have different polysomes within your cell lysate which are halted, and then you will put them on a gradient. But if you directly use the cell lysate, which has this full transcripts, and put them on the gradient, it separates the, so this is the lighter fractions and these are the heavier fractions. The RNA with a lot of ribosome on it, it sediments at the bottom of the gradient. The one which has monosomes which are lighter, they sediment at the top of the gradient. So you have here 40, 60 years, 80 years, and the polysome peak. This is called as polysome profile. But what we are looking at, what we want here is a ribosome profile. How we get that? So it's a simple alternate strategy that has been used, that is like you treat your lysis. You treat the cells, once you lyse them, you put an RNase enzyme, which basically chops off these mRNA into smaller fragments. So you have the mRNA with polysomes on it. When you digest with RNase, so you get wherever ribosome is not there, it is going to cleave off all this RNA. So you get ribosome protected RNA fragments. So these are called ribosome protected fragments or ribosome footprints. So what you do, you take this peak here. So once you do the digestion, you put them again on the gradient. So all of them now are same size. So you get all of them in single peak. So you take this fraction, you isolate the ribosome, protected RNA fragments. Basically, you do a protein precipitation and also take off the protein, take only RNA fragments that are required from this peak. And these are here the ribosome footprints. They are called ribosome footprints because these are the ones where the ribosome is directly attached to. Now, once you have these RNA fragments, these are very small fragments, like around 30 nucleotides. Unlike the mRNA sequencing, when you do, you have larger fragments. But here in ribosome profiling, you get these short 30 nucleotide sequences. And once you have them, you have to do a library generation. So you have a barcoded primers, you attach them, and so that you know you can tag them and you can sequence them. Once you have the sequence of all this, you align them to the genome browser, to the database. So you get something like this, a graph here. So this is the transcript. It's the coding region. Here, the one in blue here, this is showing the intensity of this every fragment in these regions. So you get the abundance, these are the reads, and the peak shows the abundancy. So when you do the ribosome footprinting, you always get the regions, those are only translated or where ribosome is bound to. So ribosome always starts binding at the start codon and it is released at the stop codon. So you have the reads which are from the coding region. This is a typical ribosome profile. Now, just people who ever do ribosome profiling, they just don't do only look at the ribosome profiling data alone. They also do in parallel mRNA sequencing, the total mRNA sequencing, because the information you get from both is very important to interpret the data. So here, I think you would have had this class in the RNA sequencing where the mRNA is randomly chopped. So when you take the total cell lysis, you are not isolating anything that is associated with ribosomes or anything. You are looking at the total mRNA pool which is there in the cell. So you randomly fragment it using an RNase, and again, basically the steps are same. You have the fragments, you do the library generation, deep sequencing, followed by aligning the reads to the genome. Here, it gives you the reads all throughout the transcript, basically, because you have different fragments all throughout the transcript, but these are only the fragments, those are associated with the ribosome. Now, apart from the regions which are like the coding regions. You also get information about the periodicity of the codons here. So basically when you look into, zoom in and look into the each peak, you have single nucleotide resolution. So every single nucleotide, the reads are shown here. So you get really, the single nucleotide resolution from this kind of sequencing. Now, just to give you an overview of how people interpret, even I'm not a bioinformatician, but I'm a biochemist. I find it really difficult how it is to understand the profiling and the bioinformatic analysis. But I will try to give you a simple cartoon to just get a view of how they analyze the data. Once they have the reads, they map to the genome. So how do they do the quantification? Just consider a sample pool of mRNAs. You have, within a cell, you will have different mRNAs. Some are translated, some are not being translated. So you have a pool with a lot of ribosomes. Just follow the color codes. One is with so many polysomes, so this is actively getting synthesized. There are so many ribosomes attached to it. There will be few where the process is slow. And there are few even slower than that. Then you have some where there is a pausing because the RNA is never a single strand, as the cartoons show. It is highly complex structure. It gets folded. There are secondary structures. So what happens? Ribosome might not be able to resolve those secondary structures. So it gets paused. You can study all these different intermediary situations, those are happening within the cell, to get a picture of what at that moment is happening. So once you can, when you do this, the ribosome profiling of all this and then align the sequences, you see the footprints here even are similar to the color code. If there is a pause, because of a secondary structure, you see kind of a peak here. You get more ribosome footprints there because ribosome is stalled there. And you can say that in this particular mRNA, there is pausing of translation. So like that, if there are new regions of the mRNA that is being synthesized, those also you can annotate. So those are something called short open reading frames. Sometimes there are in the huge, very long mRNAs, there are short open reading frames. So they are like basically recently they have shown that these short open reading frames, they are also synthesized. They form micropeptides, like a 10 amino acids or 20 amino acids. They are not big proteins, but these short peptides are synthesized within the cell, and they're studying the role of what these these peptides could be, because they are not detected at the cellular level because they are so small in amount, but they could be having a functional role, which is actually I'm looking at in the mouse spermatogenesis process. So next. And also even mRNA sequencing reads, you can align and you can annotate the genes which are being transcribed. And when you compare the mRNA sequencing reads with the ribosome reads, you will get an idea of what is the transcript that is highly abundant. So you have the mRNA counts. If you look at that, you will know from the mRNA that this transcript is highly abundant. Even though the transcript is highly abundant doesn't mean that the protein is getting synthesized. So the ribosome footprints gives you an idea whether the protein is actually being synthesized from these mRNAs. But if you want to look at the translation efficiency, you have to look at the both together. That's the ratio of the ribosome footprints towards the mRNA sequencing rates. That gives you the idea whether this whole protein is being, the efficiency of the translation of mRNA. So I'll just give you one simple example how people use this technique to study what is happening with the basic biochemical pathways that happen in the cell. So in the wild type cells, when this particular group who worked on this, they had a mutant which is called DOM34 mutant. So it is this protein is deleted in these yeast cells, and they looked at the ribosome footprints under these two conditions. So you see in the wild type conditions, you have the codon periodicity and the start and stop codon. Protein is getting normally synthesized. The profile looks very regular profile. But when you remove DOM34, in these cells, they see that the ribosome footprints, they start appearing at the 3' UTR region. Basically, ribosome is not there in the wild type cells. Ribosome should fall off at the stop codon itself. But they started seeing ribosome footprints at the 3' untranslated regions. So this shows that this particular protein is required to make the ribosome fall or terminate. The translation should be terminated at the stop codon. This protein is required. If it is not there, even after the stop codon, the ribosome is still continuing to translocate to the 3' UTRs, but they don't see any periodicity. It means probably there is no protein, this enrichment. You need the ribosome to be released, so there is continuous production of the proteins. There is recycling of the ribosomes, and then again translation is happening. So this is one of the examples that shows that how people are using this technique to study what is happening during protein synthesis. So every technique you use has its own strength and also drawbacks. So what are the strengths? This is highly sensitive and precise. It gives you single nucleotide resolution and what is happening at that level. And it is very precise because it talks about exactly where the pause is happening, at which nucleotide the pause is happening. And also, it gives instantaneous measurements at one particular time. What are the proteins those are being expressed? Because you need to know, even for the drug industries, to know at what stage, what protein is being synthesized or upregulated or downregulated to target the drugs accordingly. And the limitations of this technique are like, it might be having some experimentally introduced distortions because you use the inhibitor to stop. So the amount of inhibitors you use, how long you treat the cells with these inhibitors, because they are going to be toxic. If you stop protein translation for a long time, it's going to be toxic to the cell. So how you have to use them to address this question, you have to do a lot of standardization, what works for your conditions and what you're looking for. And also, because you have ribosomes always attached to this, when you isolate your mRNA fragments, you always get ribosomal RNA contamination. So 90% of your reads are always ribosomal reads. So you have to filter them to study with what you are looking for. And also, since they are short sequences, like 30 nucleotides, aligning them, the short sequences can mismatch to a lot of areas in the genome. So unlike mRNA sequencing, they have long reads, they are more specific, but when you're working with something very short reads, like 30 nucleotides, it's really, you need to be very careful when how you are looking at your data. And then the material. For this ribosome profiling, you need a large amount of cells. Unfortunately, you cannot work with the way you do single cell ribosome, single cell mRNA sequencing. So for mRNA sequencing, single cell is enough, but not for ribosome profiling. But it's always a tool. It's not a complete thing that can address everything. So you always have to work in science with multiple tools to address what you want to do. So, but this has its own uniqueness, definitely, because nothing else can address the translation efficiency other than this technique. So with this, I would like to thank you all for your attention, and yeah, any questions? Yeah.
So when you're doing this, do you synchronize the cells so they're all at the same point in the cell cycle?
I'm working with mouse testes, so we isolate the cells, specific cell type which we are looking. Like if I want to work with sperm, I take the mature sperm. So whatever stage I want to, I isolate the cells. But for some experiments, you really don't. You can also You have to look at the way the spermatogenesis, the stage. So we develop mutants which stop at that stage. So you enrich those particular cell types when you are isolating. So we address this in multiple ways, but mostly enrich the cells what we are looking for. Yeah. Okay. Any more? Okay. Thank you.